So I'm going to start. So today we're going to give you some demos about how to use some of the tools that we have developed. Specifically, we're going to focus on apertures and Biotropis. So I will give a short demo of Biotropis first, and then Minji will show you the implementation of the same sort of like workflow in apertures. And then Daniel will show how to develop apertures, like what is sort of like behind, behind the dashboard or the under the hood for apertures, and also it will guide you on how to develop your own apertures. So I don't know how many of you have heard of Biotropis or tried it before. Biotropis is a RNASeq, interactive RNASeq pipeline for people that don't have coding skills, and you can bring in your own data to the website, and we can analyze your data in the cloud. Either you can bring in your raw FASTQ file. So if you click on get started, if you have FASTQ files, you can upload them. You can also use published data. And also if you have a matrix with gene expression counts, you can upload it. So what I'm going to show you now is we're going to click on your own data, but we're going to go to Geo and search for this study that was the first study that published the SARS-CoV-2 infection in human cells, followed by gene expression of RNA-seq. And this is a paper that was published in Cell in May, but they already posted data in March, like the end of March. So this is, so typically you can get the FASTQ files by clicking the GSM file, links, but they also provided the raw data for ferrets and human in raw read counts in the table. So with this particular table, we didn't have to do too many transformations. We'll show you the table. So the table looks like this. So the genes are entrance gene symbols, and then the columns describe the experiments of the samples. And then the numbers here are the counts, the gene counts for each gene. And there are like about 22,000 genes that are listed or measured. So what you can do, you can take this file. Excel obviously is not the best way to store file. This is a text file that was just unzipped from here. So this is a TSV file. And then I have it on my desktop right here. So I'm going to click here, gene expression table. And then I'm going to drag it into this area where it gets uploaded into Biotropis. So it finished uploading and it gives you like a summary of the table. And now I'm pressing continue. It's a little bit of filling up your taxes if you've ever used like TurboTax. It's a pretty nice interface that was developed by Dennis Torre, who was a software developer in the lab. So here you have an option of uploading a metadata file, which just give you the sample name, the conditions, and the tissue, and other things that you can add for your metadata file. And it's preferred because you can have more metadata elements, or you can just identify the groups. So the groups are, in this particular case, those are the conditions. So as you can see, they have replicates. For example, for series 15, they have 2 replicates. So what I do, I just sort of like copy and paste. So we have groupings. I have to do it for all of the conditions. So it will take a little bit of time. That's the only thing that really takes time. Please feel free to interrupt if you have any questions. This is a live demo, so hopefully everything is going to go well and quickly. So this is pretty slow. So what they've done, they have different viruses that they infect this cancer long cell line. And this is the mock treatment. So those are our controls. And they have them in multiple series. So the mocks are in multiple series. And this is the SARS-CoV-2 infections for, let's see if we can just move on without doing everything. Just we'll try to focus on series 5. So let's see what happens. Yeah, it still wants assignment. So let's put something quick. This, I'm just putting it in, so we are going to ignore these samples and just focus on series 5. So I don't waste too much time. But you have, if you are using that option, you have to fill in for everything. So now we have our groups, and then I click continue. We have to wait a little bit for the next page. So it's uploading our data. And this is the next interface that gives you the different tools to select, so you can turn them on or off. You can get more information about each one. So this is a PCA, heat map, cluster grammar. So it's an interactive heat map. Also, there is this quality assurance sample analysis of how many reads were aligned, differential expression analysis. So computing differential expression. I think there's 2 methods of computing the differential expression, so you can set up those parameters later. And then volcano plots and MA plots. I'm going to only select volcano plots. And then enrichment analysis with enricher, and also I'll click on gene ontology. And then finally, we have 2 different ways to query the L1000 data. One is called L1000 CDS2. And that's an older version of this drug perturbation collection of about 100,000 signatures that we processed from the L1000 that were already filtered based on their effects. And I'm going to only pick the newer version, which is L1000 FWD. And there's some more better normalization that was done for this app. So in general, we prefer people to use this newer one. So once I selected my options of tools, now I need to select my signature. So I need to identify 2 groups that I want to compare to compute the signature. So Biotropis can only compare 2 groups. So if you have multiple groups, you have to do it, create a notebook for each comparison. So what we said, we're going to compare the mock control of the A549 long cells with the infected SARS samples. So it's 3 against 3. Typically, you want to compare at least 2, but 3 or more is better. 2 is a little bit too little, so that's not recommended. And then we are ready to generate our notebook. So this is a bunch of options that we can set for each of those tools. You can keep them as default, and then we're going to generate notebook. So now it takes about a minute to execute. And what happens, the data gets fetched into a Jupyter notebook. And then that Jupyter notebook template gets executed in the cloud. And then once it's ready, once the notebook is fully executed, we're going to get a link to that report. And that report is almost like a paper. It has all of those interactive figures, static figures, and text about the methods, and links to analysis from external tools. The only thing with Biotropis is it's what it is. So those apps that you saw, what you're going to get from those apps is very static. But with Apertures, we extended this tremendously. So you can not just do bulk RNA-seq analysis, you can do many other types of analyses, and you can create your own, you can customize things. So here is the notebook completed, generated. And then once we open it, it gets opened in NB Viewer. It takes a minute to load, but it's an NB Viewer Jupyter viewer that shows the notebook. You can tackle the code and see some of the code. So here is the original table that we uploaded. This is the metadata that we entered. You can see all those 4s that I entered to save time. And these are the 6 samples that we selected to compare. You can see that they're separated, which is a good thing. And potentially we could find some differentially expressed genes from that. And also the series, even though it looks pretty not consistent, you can see that they cluster by the metadata. So the 2 groups are separated. So the virus infected are in the green, and these are the orange are the mock treated. And then this is the sample per million. So you see, like here, you have a bunch of samples that have low reads, which is important to potentially filter. And then this is the table of differentially expressed genes. You can see that the immune genes are highly differentially expressed, and you can see them here in the volcano plot. They have very high p-values, and all of these are interferon-related genes that are a sign of infection. Very few downregulated genes. And then here you have those links to Enricher, and we're not going to click on them, but this is also using Enricher. We fetch the information from the signatures that we care about, the biological processes, comparing the up and down genes. So the up is red and the down is blue. And whatever is highlighted in bold, those are the significant terms based on their enrichment p-value. So for example, in response to interferon has a very low p-value of the corrected p-value is e to the negative 27, which is high significant. And you can see the genes, 30 genes that are upregulated that overlap between the term and the upregulated genes from the actual experiment. So this is finally the drug mapping the signature onto the L1000 data for finding up or down regulated genes. And then here you can see that we get some blue areas and red areas. And then those are drugs that either reverse or mimic the expression. So the red are the mimickers of the signature. They are turn on immune response, and the blue ones are suppressor of the immune response. We know that this area is NF-κB suppression. The drugs that suppress the NF-κB pathways are anti-inflammatory drugs, and those are inhibitors of cell cycle. And then we can get the actual results. You can get them in those tables. So these are the top drugs, the similar signatures and the opposite signatures. So before we proceed to Minji, I wonder if anyone has any questions at this point. I'm gonna stop my sharing.
So I have a quick question. Since me and probably a few others were assigned to W1 with some delay, is it possible very quickly you explain the initial couple of steps? Just if you could spend a minute, that I'd really appreciate that.
Sure. So I'll just summarize. So we use Biotropis, which is the app that you saw that we went through. And to use it, we first identify a study from Geo, and we took the
Do you mind to share your screen again? Sorry, couldn't see your screen.
Yes.
I also joined very late, so I actually didn't hear the talk either. Sorry. Like they switched very late, so I literally saw the very end.
No problem. So what I showed is that we went to Geo, we zoned into this study that was the first study that profiled SARS-CoV-2 infections in human cells. And then we downloaded this particular file, the human raw read counts from here. We unzipped it and then uploaded it into Biotropis. And then going through the steps of Biotropis, we went through the process of uploading the data, annotating it, selecting the tools that we want to have processed, and then we generated a notebook that was a report of the results from that study. And that it gives you like almost a paper that analyzed that table of data with PCA plots, heat maps, different enrichment analysis, as the last thing is the querying against the L1000 data. So this is an app that was already published at the end of 2018. And since then, we have advanced our work, extended this to create this Aperture project that Minji is going to talk about next. So I'm going to stop my sharing again.
Hi, I had a question. A great talk. And I wanted to know, so the results that you get from Biotropis and the result that the paper published, how do they compare? If that makes sense. Like
yes, it's most of the time they compare. Like in this particular case, we got also interferon response. The paper highlighted the fact that in some cases, specifically in human tissue cells that from like human lungs and ferrets, there is a suppression of the interferon response. Their claim that we only see cytokine response, but we don't see the interferon arm of typical immune response. We don't see it here in those cells, and they did not see it in those cells either. But if you do the same analysis and you're looking at the human lung samples from patients that they have this in the study, you'll see that it is mostly cytokine response. It can be different because we may use like a different method. We use Lima for processing the data. We may be using different normalization method. There are some options. So if you don't get agreement, you might want to go to the methods of the paper and see what normalization methods, what tools they use. In some cases, it could be that this is there's a shortcoming in the way we process the data because it's not like a size-fit-all solution. In some cases, the people that process the data from the study, they may have missed an important step, and our results may be more robust. But that's a good question.
Yeah, thank you. And is it possible to like tweak the code in the Biotropis Jupyter Notebook?
Tweak the code? Yeah.
As in like, if you want to like add some like command lines inside the code, like.
Yeah, so that's a great segue for Minji's presentation because with Apertures, it's much easier, but in general, you have to download the notebook, and the notebook is available for download, and then you have to execute it. Like, so you can click here, download the notebook, you'll get the code, and then you could potentially run it yourself locally. Okay. But with Apertures, it's easier.
Okay. Thank you so much.
Okay.
Excuse me, I have a question.
Sure.
I was wondering about the Jupyter, no, sorry, the BioJupy, whether you can import, for example, RNA gene expression microarray data as well, or is it specifically for the RNA-seq data?
It's specific for RNA-seq. So for microarrays, we have a app called the GeoAutoEnricher, and that app is geared to processing microarray data. I think you could upload, you can try it, but I think it's a little risky because the algorithms and the normalization methods are geared to doing raw counts for genes.
I see. Could you please type in the chat the app
Yes.
you mentioned for the microarray data?
Yes.
Thank you.
Okay, can you see my screen?
Yes.
Okay, hi everyone, my name is Minji. I'm going to introduce two Apertures today. Aperture is a web tool that converts Jupyter Notebook into a biological data analysis tool. I'm going to show you the Burke RNA-seq analysis Aperture and the Germanism consensus terms Aperture for analyzing COVID-19 datasets. First, I would like to introduce Burke RNA-seq analysis Aperture. Gene expression profiling of Burke cells with RNA-seq is now the most common method to profile gene expression. So it is necessary to develop computational analysis pipelines that can glean more information from such data. Therefore, we developed Burke RNA-seq analysis Aperture that enables non-computational users to analyze and visualize their RNA-seq datasets. The Burke RNA-seq analysis Aperture includes data normalization, 3D interactive plot of samples, and hierarchical clustered heat map, and differential gene expression analysis, and small molecule identification. And the URL is on the top, so you can try to run it with your own data sets. To run the Aperture, first you need to prepare RNA-seq samples. I'd like to use this COVID-19 dataset, which was published in the last July as an example. This dataset includes expression values of infected cell lines treated with several drugs. And we are going to see which genes are differentially expressed after treatment in SARS-CoV-2 infected cell lines using the Aperture. As an example, I downloaded 3 berberine treated samples and 3 GMSO treated samples. After you download the samples as you want, and you should generate 2 files. One file contains expression values of samples, and the other file contains metadata of the samples, and save them as a comma-separated or tab-separated formats. Then the Burke RNA-seq analysis Aperture starts with an expression matrix and metadata of the samples. You can upload 2 prepared sample files. And then you can specify parameters for your analysis as you want. For example, the Aperture implements various data normalization methods, such as Z-score normalization and quantile normalization. So you can select normalization methods. And you can also select visualization methods among PCA, TSNE, or UMAP. Moreover, you can manipulate parameters for differentially expressed gene analysis, such as thresholds for the number of genes to be analyzed by enrichment analysis. After you submit, the analysis results are visualized using various plots. The first figure displays an interactive and three-dimensional scatter plot of the data. Each point represents an RNA-seq sample. Samples with similar gene expression profiles are closer in the three-dimensional space. In this example, the control samples treated with DMSO are in blue dots, and the case samples treated with berberine are in orange dots, and it looks like they are clustered. And the figure on the right contains an interactive and hierarchical cluster heat map using cluster grammar, and it displays gene expression for each sample in the RNA-seq dataset. Every row of the heat map represents a gene, and every column represents a sample. And you can explore the similarity between samples and genes in an RNA-seq dataset. And gene expression signatures can be quantified using differential gene expression method, which compare gene expression between two groups of samples to identify genes whose expression is significantly altered in the perturbation. Volcano plots are used to display the result of a differential gene expression analysis. Each point in the scatter plot represents a gene, and the axis display the significance versus fold change estimated by the differential gene expression analysis. When you hover over it, you can see more information of genes. As a result of differential gene expression analysis, we also provide enrichment analysis results. Enrichment analysis is a statistical procedure used to identify biological terms which are overrepresented in up or down gene sets. These include signaling pathways, molecular functions, diseases, and a wide variety of other biological terms obtained by integrating prior knowledge of gene functions from multiple resources. To do this analysis, the Aperture uses Enricher, developed by the Mayan lab, to perform enrichment analysis using a large collection of gene set libraries and various interactive approaches to display enrichment results. For the COVID-19 datasets, you can see the cholesterol-related gene ontology terms are enriched in the berberine treatment samples. L1000 FWD, which is also developed by the Mayan lab, is a web application that provides interactive visualization of over 16,000 drugs and small molecule-induced gene expression signatures of LINKS project. Signature similarity search is implemented to enable the search for mimicking or opposing signatures given as input of up and down gene sets. For COVID-19 data analysis, small molecules that can mimic the gene expression signatures of berberine can be another potential COVID-19 treatment. Germanism consensus terms is the second Aperture to be introduced today. Let's say we have a collection of drug sets and we want to find the consensus enriched terms given a collection of drug sets. This Aperture performs drug set enrichment analysis for all uploaded drug sets against drug set libraries. The top consensus terms are visualized using stack bar plots and heat maps. For this Aperture demo, I'm going to show you the analysis using COVID-19 drug sets. The Mayan lab collects the COVID-19 drug and gene set library from various resources, and the URL is on the top. From the library, we can get 12 COVID-19 drug sets in GMT format as shown on the right, and try to find enriched terms among those drug sets. You can upload the GMT file and select drug set libraries that are from Germanism. Germanism is a collection of libraries of drugs, and each library is curated from a unique resource that has information of drug attributes. For example, drug attributes include gene ontology terms of gene signatures from drug perturbation profiles, or targets of drugs or substructures of drugs. You can select libraries as many as you want. And in this example, I selected L1000 FWD gene ontology libraries to find consensus enriched terms of gene signatures from drug perturbation profiles. As a result of the analysis, you can see a stat bar chart of enriched terms sorted by enrichment scores. One of the things to highlight is that gene ontology terms related to regulation of cholesterol metabolism and synthesis were significantly enriched across the 12 independent drug sets. It has been reported that patients with high cholesterol and hypertension are at a higher risk to develop COVID-19. And previous literature reports that cholesterol has important functions in regulating immune function. Furthermore, recent literature supports the idea that cholesterol-lowering drugs may induce the severity of COVID-19. In conclusion, I have introduced two apertures with examples for COVID-19 dataset analysis today. Using the apertures, you can get enrichment analysis between two groups, and you can find potential drug candidates. Moreover, you can get top consensus terms of collection of drug sets. We could identify cholesterol-related mechanism is enriched among the 12 COVID-19 drug sets. Okay, this is the end of my presentation. Thank you for listening.
Thank you, Minji. Maybe if anyone has like a burning question for Minji, we don't have a lot of time. We hope that we're not going to get cut off like yesterday. But do you guys have any questions for Minji before we go to Daniel? All right, I guess we should go to Daniel. So Daniel will show you how to build apertures, what's happening behind the scenes of all of those apertures. And we currently have those 40 apertures that can do various types of analyses. And also, yeah, you can contribute your own, if you have your Jupyter Notebook workflow and you want to share it through here, there's a way to do it.
Okay, so I'm going to try and speed through this because yesterday they just cut us off like on the dot. So, but here's kind of the catalog, the Aperture catalog. We'll share this. This is pretty new, so we're still in the process of developing it. But part of this, it's making a new paradigm so that these types of applications that Minji just showed and BioJupis as well can be created pretty quickly. And because of that, I'm going to essentially walk you through a little bit of how you might be able to create your own. After installing the library with from our GitHub, you can also view that GitHub and see the documentation there. There's information about how to use it and extensive generated documentation on the available fields. For the purpose of kind of this demo, what I wanted to go over is a gene transcript conversion aperture. So this was actually mentioned during one of the talks. How can you go from, say, ensemble IDs and then get regular genes out of it. And so for this purpose, I designed a Jupyter notebook that could do it. Basically, the NCBI Resource has an FTP server. Where you can actually download these gene info matrices. And that's kind of where this starts off. So after importing like some basic NumPy and Pandas tools, we can actually load this resource for humans. And once we load this, we get a table that has kind of entourage gene symbols, the gene ID, and all types of synonyms and external database references, map locations, and more. And so to kind of have a case study, here's some COVID geostudy that I found that, you know, when I download this data and look at it, I've already downloaded it, and it's over here. And so, oops, did it twice, but so here is that data, right? So we have ensemble IDs on the index and some samples on the columns. And so in order to do this lookup, we can take this NCBI gene table and kind of take all of the symbols, the gene IDs, synonyms, other designations, process them to create this kind of like lookup table that goes from the actual some synonym and produces some official symbol, right? And so this ensemble ID will go to this particular symbol. And when we do this, it turns out that in fact there are duplicates when you do this. And so for the purpose of this, I'm just going to drop these, but a more extensive look at this may come up with more creative ways of getting rid of these duplicates. But once we've done that, we have a disambiguated lookup table. Now, in order to apply our lookup table to get this conversion done, what we can do is essentially map all of our ensemble IDs to transcript genes. The problem with just doing this is that in some cases, you may have some transcripts that map to the same gene. And so to overcome this, what we do is we'll take the variance of all of our transcripts, and we'll use the one with the essentially the highest variance, and then we'll use that gene. And once we've done this, we end up with kind of this new table that has become shortened, right? So we had a bunch of transcripts, now we have a bunch of genes. You will notice that this includes some non-coding genes and other types of things, and we'll address that soon. So once we've done this, we have a way with Jupyter to actually do this, but to turn it into an aperture, what we need to do is add some annotations. We just include this top part here, and then what we do is we can declare different fields. In this case, I declared a file field that allows a user to accept a file. And once we do this, we can actually use the variable that the user will provide on the form to actually render code. And so if I actually run this locally, transcripts genes, looks good. Sorry, I'm trying to rush too. In the wrong directory.
They did give us 10 more minutes, so we will get cut off. We'll get cut off at 5:45 exactly.
Okay, that's better. Okay, so like this, of course, because I wanted to get here, I rushed through this, but basically this terminal application for apertures, where we just use the aperture CLI and give it the notebook of question, we have some alternative options which can make our output a bit prettier that are described better in the documentation. But the idea here is that our actual Jupyter notebook, once we've specified, hey, we need a file here, and that's going to affect this code. And in terms of this, like NCBI gene information, basically, I just took human for that particular experiment, but people may want other organisms. And for that, I went through kind of which organisms are there and created a choice field that points to each of the individual organisms that a user could select. And then like the types of genes that they want included in the conversion. So maybe you just wanted the protein coding genes. And so this filter will get applied once they've selected this. If I have to have this running. So once they've selected this, like this gets substituted. So our example was Homo sapien, and here was protein coding. But if the user, for instance, selected also ncRNA, then this will also change. But this is still running. So another thing to note is that once I like save this, this updates in real time, right? So I can really do this development right away. But the idea is once I've built this Aperture, then I can actually run it over here. So you can provide, say, this example file, the exact analysis we did before. When we click submit, it actually just runs through this Jupyter notebook and produces a output in real time for some external user, showing all the intermediary steps and then getting their conversion. And now basically, once you've done this conversion, the user can download the output, which would be this table here. Furthermore, these are like persistent identifiers, so someone else could come back to retrieve it. And so once we've made this Jupyter notebook, effectively we have an application that people can come in and bring their own data. They can browse for their own data, or actually customize what organism and things like this. We can further add more options. Apertures even gives us the ability to do things like if statements, and more. So basically this magic permits you to do this. All right, and so let's see. Okay, I don't know how that works, but it should work. Yeah.
So perhaps we can show how to submit an Aperture for like a pull request, and then we should open it to questions because we don't have a lot of time.
Yeah. So after that, basically, given that you have kind of your Aperture, you can basically just specify the Python requirements that are necessary to actually execute your Aperture, put them into a directory, and then you can actually contribute these to join the Aperture catalog on our GitHub here. And you'll see that most of our Apertures are all here in their own directory. Each Aperture will have this basically a README describing the Aperture, the actual Jupyter notebook for that Aperture, their dependencies, and an Aperture JSON that basically just has some other metadata like the version, the title, some image if you have one, who authored it, things like this. And you can also kind of look at some other pull requests. People have been submitting Apertures for a while. We've been using it internally in the lab to facilitate this. So definitely, if anyone has any questions, shoot, give them so we can answer them before we get cut off.
So I guess I hear quiet. So I don't know, maybe you can show, go ahead. I hear some beginning of a question. Maybe not.
And anyone who's interested in this particular Aperture will probably publish it in like next week on the catalog.
Yep. I think it would be nice to add a few more conversions. Like, for example, I just looked at one study that provided gene IDs.
So this will work with gene IDs. It'll work with, so basically anything in here. So HGMCs, okay. MIM, synonyms of genes, locus tags, if they're provided. And maybe we could even make them work for chromosome locations. But essentially all of these different other designations and things like this are all added to this table to do the lookup.
I guess you could show them the enrichment analysis with the scatter plot Aperture. So there is some documentation also on this site.
Yeah, so the idea here, using a similar, like the default list that Enricher has, we can actually visualize it within a particular background. So I'll leave the default. But we have a bunch of other backgrounds. You can pick some significance values. And once we do this, just like you saw before, this gets executed. And once it's done, we get this type of scatter plot, which is essentially showing your enrichment results colored here, the significant ones, in the context of the actual different terms that were in that library. So we can see that almost as expected, our enrichment results are clustering together to an extent. And then we can hover over them and see what they are. And this is just one of many possible ways we can augment enrichment results or other types of data analyses by building a Jupyter notebook and then deploying them as an application that people can insert their own data for.
Thank you, Daniel. So I think we are done. I think thank you so much for attending, and you can always like just contact us with questions by email or GitHub.
I see Marissa asked a question. Biotropis and the bulk RNAseq Apertures seem similar. When do we use them? Yes, they are similar by design, I think. The kind of bulk RNA-seq Apertures was one of our first attempts at like recreating Biotropis almost within the confines of the Aperture to make sure that Apertures were flexible enough. Biotropis is certainly more battle tested than Apertures at this point in time. So I'd definitely say if your analysis works with Biotropis, I would use that. Apertures currently don't have a way to convert FASTQ files into matrices either. But if you did have you needed to convert these transcripts, BioGPs doesn't do this. Once we've released this Aperture, you potentially can utilize it to get your matrix to put it into BioGPs.
Also for Marisa, one of the things that we've done, even though it looks very similar, what Minji did for the bulk RNA-seq Aperture, she brought in the code into the notebook. So with BioTropis, it's a little hard to trace the code because it's all made of libraries for each section of the notebook. So Minji made it more transparent. So if you are interested in modifying the notebook and it sort of tracing exactly how the analysis is done, I think the Aperture version is more transparent. Regarding Biomart, so Biomart is a good way to convert, like they have a nice ID conversion tool, but it is a little bit complicated. You have to download it and install it. It works locally. And with the Aperture, it might be better for non-experts. And yes, I think one of the enhancements to the Aperture that Daniel created in like one day is to also add transcript to gene conversion, which is pretty straightforward to do. We do that a lot. And now with Apertures, we can make that available for novice users that need that task like pretty frequently, but don't really have the time or the skills to go through and figure out how to run things from the command line. Okay, thank you so much, and stay in touch. And I guess goodbye. It was a pleasure to have this workshop today, and the next meeting, I think it went really well, and that will be it for links. That's the last big meeting for links. So have a great weekend and stay safe.